with the telomerase enzyme similar with the standard drug, that is curcumin. The PLP score of 58.55 for BNF and curcumin for 68.55. So this is the summary. Just 30 seconds. 30 seconds. Sorry for the intro. So what is my aim when we are inhibiting the target-based enzymes? Like as yesterday, Roy sir was explained that IDH1 if we inhibit, then automatically glioma will be inhibited. Like that, the enzyme which is a telomerase, which is not present in the normal cells, but only present in the cancerous cells, which is more actually involved in the cancer progression. So it is useful for the diagnosis as well as for the treatment. So the VNF, which is the synthetic flavonoid we used here, actually inhibiting the telomerase activity, which initiating the inhibition of proliferation and trigger the senescent pathway and initiate the senescence at a earlier stages, and cell death will be happen. So means conclusion is, when we are getting a specific area, then automatically we can avoid the side effects. Those side effects minimized and reduced, efficacy will be increased. Thank you so much for giving me this opportunity. Thank you for organizers for giving me this opportunity. Sorry for the interruptions earlier.
At least some basic questions also you can ask, because he has done the work in UK, Mr. Dr. Mahinder.
Yes, but this work is done in biotechnology department, but earlier the published paper is in UK. Okay. This is here itself.
What is the role of P53 gene?
P53 is a one type of tumor suppressor gene which is mutated in more than 65% of cancers. Means here also in cervical cancer also it is mutated. If tumor suppressor genes are active, then there will be no cancer. I will give you the guarantee. If your tumor suppressor genes of the tumor suppressor P53, if it is not mutated, if there is no abnormality in the tumor suppressor gene, there is no such type of transformation of normal cancer to non-cancerous to cancerous cell will not happen. Why? Because it stops in cycle. It gives the time for the repair. If there is a repair is happened, then it opens the break at cell cycle checkpoints. Then the further stage of the cell cycle, if not, it arrested the cell cycle. Automatically, there is no probability such type of abnormal cell will be present, but the death of the such type of cells will be happening. Without abnormal proliferation, that entering into the, when they are in the benign condition, think that this is a cancer, then it converted malignant, when it converted into malignant, then only you have the problem. Okay, more than 500 diseases are cured by the curcumin, not only inflammation.
That's what I know.
Okay.
Inflammatory compounds have therapeutic effects on that.
Yeah. Okay. So we did the in vitro analysis, in vitro study to validating the third, which is a telomerase active component, one of the cancerous cells only, not non-cancerous cells. We have the result in vitro conditions, but to validate it, whether it come in contact with the telomerase or not. What is the mechanism behind it? To validate that, we did the molecular docking studies and we validated that there is an interaction with the parts. If there is no standard, the comparison will be not done properly. That is the reason why we took curcumin as standard. Why? Because there are so many publications, more than 20 to 30 publications, curcumin which inhibit the telomerase. So that is the reason why that took as a standard. More than 2 to 3 papers also related to the standard.
There are any more doubts? I think students have, so free time you can discuss with us.
The topic is related to CRISPR-Cas9 mediated functional analysis of RAS oncogene, which are homologous in Drosophila melanogaster compared to human studies, Implication for colon rectal cancer. Whatever mortality cases, top 3 cases reporting in the colon rectal cancer nowadays. Colon rectal cancers arises from the abnormal epithelial cell growth into a colon, in the colon and rectum, which are having the mutations in the genes like RAS and APCs. So RAS oncogene in cancer biology, KRAS, NRAS, HRAS, these all encode for a small GTPase. KRAS, NRAS, HRAS, these all encode for a small GTPase. Those binding protein act as a molecular constitutive activation and uncontrolled like so CRISPR has different stages to different considered region of the RAS64 gene for precise Cas9. Then Cas9 mediated cleavage occurs. Cas9 nucleus introduced a double-stranded break at a RAS64 targeted site, repair pathway selection, then go for a micro injection like CRISPR component delivered directly into a Drosophila embryos for germline editing, mutation validation, PCR amplification and sequencing confirming successfully edited for the RAS64 locus. Gene expression, total RNA extracted from the Drosophila melanogaster, providing the RAP also for the downstream expression analysis. cDNA synthesis were also done. Key results, sequence analysis, which have high similarities between human and Drosophila. Phylogenetic analysis, RAS64 and human oncogenes were clustered closely. PCR amplification, targeted RAS fragments amplified successfully. CRISPR editing, targeted mutations are also successfully introduced. Gene expression, altered RAS expression is edited and controlled samples were there. Functional outcomes, mutated RAS induced cancer, the phenotypic changes occurred. And topic discussion. Conclusion, conserved homology, CRISPR-Cas9 successfully implemented and cancer pathway role validated model. Future, we can also do like yesterday man said, precision medicine. Precise medicine can be done for the particular patients for this and drug screening, future studies, omic integrations also done. And also RAS64B mutations in depth define a specific role in the cancer, to the colon cancer, there will be a regulatory notes also. Thank you.
Now the session is open for questions.
How did you design primer?
Primer designing generally by two process. Most of the people use primer 3.2, or we also can do in NCB by direct using blast technique. We can do primer designs.
The one you have not used, the experimental pictures.
Ma'am, actually, experimental pictures, I really thought we should mention it. That's the reason I gave it. Actually, it's under now it's under review of my guide. If it's done, I have to go for completion.
Next presenter, B. Suresh.
Lakshmi.
Good morning to one and all. My name is B. Suresh. I am from Palmur University, Department of Geology. Today my topic is about accumulation of heavy metals in Musi River water and their impact on fishes. And introduction of these heavy metals or the metallic elements characterize high atomic weight and density, excluding 4 grams per centimeter square. Commonly, heavy metals include chromium, manganese, lead, zinc, cadmium, and copper. And while trace amounts of some of these metals are essential for biological functions and metabolic activities in humans, and elevated concentrations can lead to a range of acute and chronic health disorders. And heavy metals are contaminated in the Musi River mainly by natural sources and human contamination. But natural sources are atmospheric deposition, hydrothermal events, sedimentation, resuspension, weather of rocking, volcanic activity, and soil erosion and forest fires. By human activity, industrialization, urbanization, electronic waste, vehicular emissions, construction waste, and agriculture practices, and contamination of fishes with heavy metals. Fishes are highly vulnerable to heavy metal contamination because they are widely found in aquatic environments and act as the bioindicators of the water. And heavy metals are non-biodegradable, can lead to long-term toxicity through bioaccumulation and biomagnification in tissues. And metals enter fish through gills, skin, digestive tract, and contamination of food. Objectives. There are mainly three objectives: to assist heavy metals in the Musi River water and to evaluate toxicological effect of heavy metals heavy on fish psychology and to provide recommendations for pollution control and fish consumption safety. Methodology. Fish samples are collected from Musi River in Solipet village and Ketapalli Mandal and Nalgonda district. These are mainly I have collected Vicus gillus. It is commonly known as Viscox catfishing and native name of Telangana is Jella. The samples are taken in the month of June 2025. The flowchart of methodology: sample collection, waterman's filter paper 100 is taken for separation of solids and measurement temperature sample, acid digestion at 20 ml of aqua regia. Aqua regia is the percentage of 3:1 of HCl and HNO3 and heating. By using heating, temperature can be maintained at 90 to 95 Celsius and duration to 2 to 4 hours. Analysis: the test of chromium, magnesium, cadmium, zinc, lead and copper. Method used for atomic absorption spectroscopy. So this method, the specificity of material, how much substance heavy metals can be there, it can be useful. And in the lab we have calculated the bar. Flowchart: first sample sites are Musi River collection of fish samples. Formaldehyde injection is given to them in the mouth for the preservation of it, and storage of labeled contaminators, transport to the laboratory, dissection and washing and drying of sample, acid digestion process and heavy metal detection. Fish samples were collected from Musi River near Solipet, Ketapalli Mandal and Nalgonda. These selected species are commonly consumed fishes, namely Micus gario, long viscous caddisfish. These samples are taken after collection. Immediately each fish is preserved by injecting 10% of 50 ml formaldehyde solution in the mouth by using syringe. This method ensures preservation of internal organs of and tissues, preserving early decomposition. The preservation specimens are stored in tin and labeled where the stored and containers are contained. These are the results according to WHO. These are the WHO record readings, and in this diagram, in this chart, the chromium is highest mean and standard deviation, and the lowest is copper. Cadmium is the lowest, and according to pie chart, the chromium is more, and the cadmium is 1%. And these are the problems that occur in the heavy metals in the gills. On the top point is very important because the slightly bending of primary and secondary gills lamina and heavy metals that affect the liver. One hepatocytes are discoloration is observed and necrosis, no necrosis, and fat degeneration and swelling and vulcanization, and minor variations of cell species or phytoplasmic staining likely due to natural tissue variation and sample division. Minor irregularities in muscles and fiber alimentation is observed. Conclusion: provides important data on heavy metal accumulation and water. Scope limited to selected sample location and fixed sample. Seasonal changes in contaminated not evaluated. Long-term impacts and fish psychology, reproduction and ecosystem health remain unclear. Investigate a broader range of metal, includes multiple water bodies and seasonal sampling. Findings support strong monitoring, prevention and I will give few suggestions. One is routine monitoring, development of site specificity, safety standards, and improved industrial waste management, strengthening regulatory frameworks and community awareness and state food preparation. Further research and bio monitoring studies are required. Okay, thank you.
Any questions?
What
Can you
is,
know that,
madam, one question. What is asthma regulation?
It is maintaining of temperature, body condition maintenance.
Body temperature.
Regular according to seasonal variations, maintaining of temperature.
Have you studied any behaviors? Have you observed any behavior? Have you done benchmark?
Yeah.
Have you observed it?
Experimental.
This is experiment. These are my key experiments. which local name is Jella, and we have collected it in a Solipet. Here, Nalagonda district, in Nalagonda district.
Okay, which part of you collected from them? The sample?
Total fish collected and we total fish.
Real muzzle.
One part we separated did experiments after collection in the laboratory.
There is the fluoride content in the water.
Yes, ma'am.
Is it? It has any effect on the fish?
No, ma'am.
Among the two methods, bioremediation, phytoremediation, which is the best method?
No.
Among the two methods, that is bioremediation and phytoremediation, which is the best method for removing of heavy metals from the contaminated water?
No idea.
Okay.
How can we calculate bioaccumulated fish?
Bioaccumulation. In bioaccumulation is the heavy metals which are taken by the contaminated metals are stored in the fatty tissue rather than its elimination.
That is okay, but which technique you are using?
Not understood.
Injected 50 ml of formaldehyde. For that, afterwards isolated tissue or not?
Sir,
50 ml.
50 ml formaldehyde. 10% of 50 ml formaldehyde used.
Then all the body parts are swollen or not?
Some
3 ml, not 5 ml or 2 ml. The dose will be 0.1 ml to 1 ml to 2 ml. 3 ml like that only. This is also you can check that.
Okay.
50 ml. Body fish weight is up to 10 to 15 grams only.
Okay.
Small questions.
Okay, thanks.
Next participant. Monica. Monica, you are next participant.